Welcome, Mindsetters, to Learn Extra Live Bootcamp. Today we're doing life sciences. This next to me is Aslam. Hello, Mr. Aslam. How are you?
Good, Pat. How are you doing?
Great, thanks. What are we going to be doing today?
Today we're going to be dealing with DNA, RNA, protein synthesis, and meiosis.
Awesome. I'm sure you guys have plenty questions to go through today. So Aslam is going to be here to answer all those questions that you have, as well as some education that you guys may need. So while I prepare them for the show, you can just prepare there. So guys, you know that with the boot camp, there is lots going on, and you know that there's also a challenge question. This is the competition question for the show today, and you stand a chance to win some awesome prizes. Without further ado, let me tell you what the question is for today. In DNA, thymine always pairs with A, adenine, B, uracil, C, guanine. So that's in DNA, thymine always pairs with A, adenine, B, uracil, C, guanine. So you guys can send in your answers throughout the show. There's also a unique code. Guys, don't forget the unique code when you answer the question. The unique code is 1504. Put it right at the beginning so you don't get strayed by anything else. Also, very important, you must answer on the events wall and not the actual wall. Any questions that you answer on the other wall, that may be lost in the cracks. So please answer on the events wall. Another way you can answer is to email us. Email win@learnextra.co.za if you don't have Facebook, and if you do, also, you can SMS us. You know our SMS number. Let me read it out to you. 083 44 888 10. We also have PepText. PepText is free, guys. So download it on your phones and send us everything that you may have. You also can ask us any questions today, because it is a boot camp and we want you to be squeaky clean for the rest of the year. Over to you, Aslam. You can take it away.
Thank you, Pat. Just to remind you, the question again. In DNA, thymine always pairs with A, adenine, B, uracil, and C, guanine. Keep that at the back of your minds. Write it down if you have to, and send your answers in as Pat has requested. Guys, while we wait for your questions to come through, remember, we are very interested in getting your questions so that we can deal with the problems that you are having. However, while we wait for your questions to come through, we are going to go through some of the questions that we have prepared for you. And as usual, wherever we go, people say that they have problems with protein synthesis. So today we'll kick off with a question on protein synthesis. This one here is adapted from March 2010. Paper 1, question 2.2. The question says, study the diagram below, which shows part of the process of protein synthesis. Let's look at that diagram. There you have the diagram. There's 4 letters, A, B, C, and D. And they have the number 1 in that area as well. Let's look at the questions. The first one says very clearly, provide labels for structures A, B, and D. And I'm going to do that on the diagram so it makes life easy. A is pointing to this little envelope in this area here, separating this area from, from this area here, this area and that area. This is a barrier between that. So A is the structure, and that is the nuclear membrane. The nuclear membrane. Good. B, let's look at B and D at the same time. And this is in itself something to explain to us the difference between these two molecules. Let's look at B. Look at the letters that are found there, U, G, A, and C. Those are the 4 different nitrogenous bases that are found there. And look at D, has A, C, T, G. Those are the 4 nitrogenous bases found in D. At the same time, let's look at the number of strands found in D and those found in B. D has 1, 2 strands, while B has 1 strand. So guys, those are some of the differences between these 2 molecules. And obviously then by now you would have guessed that B is RNA and D is DNA. Why? Because D has 2 strands. At the same time, it's a double helix, a spiral molecule, as you can see. And it has this special feature here, thymine. It has also adenine, cytosine, guanine, but it has thymine. Notice on B, there is no thymine. Instead of thymine, there is a U, which is uracil. So that is why B is the RNA and D would be the DNA. Please understand that. That's for the first question. The second question says, state one function of molecule D. Remember when you're answering a question like this, where they say state 1 or state 2 or state 3 or state 4, whatever it is, the first 4, or in this case here, the first answer is considered. If you give 5 functions and your first one is incorrect, you get 0. That's it. You will not be credited because the markers are instructed to mark the first one only. So they want to know a function of D, and you have labeled that as DNA. So what is the function of DNA? Yes, you got it. Already from grade 10, you should know this. And the answer is it contains hereditary material. And we know by now, if you are in grade 12, you'll know that this hereditary material that we are talking about are known as the genes. No, not the genes that you wear, what Pat is wearing at the moment, or what you're wearing at home, the genes that make you. G-E-N-E-S. These are the codes that give you your characteristics, your unique characteristic, the color of your eyes, the size of your nose, the size of your bum. Sometimes, however, remember, it's not only the genes that make up the size of your bum. Remember that, or the rest of your body.
What else is there?
That's all putting all those unhealthy stuff into your
McDonald's,
body and not being active. So guys, remember, be active all the time. Good. So that is the main function of DNA. However, we do know, because we have studied about DNA and we are doing protein synthesis, DNA also codes the message for the formation of various proteins. It provides the code for the formation of different proteins. Okay? Which part of protein synthesis takes place at 1? Let's go back to 1. 1 is in this area here, and specifically here something is happening. And if you look closely, you'll notice that DNA nitrogenous bases are exposed, and alongside something is forming. And this something has U, as we discussed earlier. If it has U or uracil, it cannot be DNA, it has to be RNA. And because that was the nuclear membrane, and anything inside is the nucleus, then this would be messenger RNA. Because messenger RNA is the only one that is found, only RNA that is found inside the nucleus, and it goes out of the nucleus. Okay, so that's messenger RNA, and messenger RNA already should trigger another thought in your mind, and that is that messenger RNA in the nucleus is involved in, you got it? Transcription. Good. Let's just go back there to remind ourselves about this transcription. You've already gone through the recordings and all the other things that you have learned, your textbook, your workbook, etc., and you've learned about this transcription. Just a hint. transcription. Simply means to transcribe, to make a copy. So transcription is that process where messenger RNA copies the code from DNA. And in the process, obviously, messenger RNA is formed. This is transcription. Where does it take place? It takes place in the nucleus. What provides the code? DNA. How? It acts as a template. And what does it produce? It produces messenger RNA. What does the messenger RNA then do? It leaves the nucleus. As you can see, there's one there. There's your B going out to organelle C. Good. Name the type of proteins that control the process named in question 2.2.3. This is something that you're carrying with you from grade 10. Every chemical reaction in your body is controlled by this special type of protein, and that is enzymes. Remember, when you studied enzymes in grade 10, you would have said one of the properties of enzymes is that all enzymes are protein. All enzymes are protein. So this is, but at the same time, the fact that they are protein, they also control these reactions that lead to the formation of protein known as protein synthesis. When we spoke about DNA replication, we also said that enzymes control the process. The next question says, identify organelle C. This is where this fellow, our friend, messenger RNA, is going to go to organelle C. Obviously, anywhere here is obviously the cytoplasm. But the cytoplasm is not an organelle. They want to know specifically, name an organelle. This is where we must be very careful. Organ, tissue, cell, organelle. We're going smaller. From the body, organism is made up of many organs. These organs are made up of tissues. The tissues are made up of cells. And inside the cells, those structural things that have functions are known as organelles. So in the cell, there's an organelle. And this organelle is known as the ribosome. So that's the organelle that we're talking about. Name and describe the part of protein synthesis that takes place at organelle C. But by now you should know the name of this process, because we've already eliminated one of the two names that are associated with protein synthesis processes. One was transcription, and the other is translation. And this question is asking for organelle C, and let's go back to the diagram. We said that here transcription is taking place. So obviously at organelle C, at the ribosome, we have the other one, and that is known as translation. So let's discuss that. So they said name and describe. Now guys, I find this when we mark in as well. Although the question said name and describe, many of you just go into the process. Now if you did not name it, you know the process, or you know the name of the process, but you have problems in describing this process. At least the first thing you do is you look at the instruction. The verb says name. So you do that. Name, translation. Okay? So that's already giving you 1 mark. But we need a little more than that. They say describe. And whenever they use the word describe, explain, discuss, suggest reasons for, we need an elaboration. So an explanation is required. So this process of translation is taking place at the ribosomes. How does it take place? Starts with messenger RNA, attaches to the ribosome. And this attachment is just not haphazard. This attachment in a specific way. How is this done? Such that it exposes, or exposing, I'll write here to make sense, exposing the nitrogenous bases, and so forming what we call the codon. So this messenger RNA bases, the messenger RNA bases form the codon. And when that happens, the transfer RNA molecules pick up amino acids from the amino acid pool. By the way, this is not a swimming pool that we are talking about. It is the area in which amino acids are found. It's not a specific name, meaning there's a pool of these things, their availability. From the amino acid pool, where in the cytoplasm, and brings them to the ribosome. The transfer RNA attaches to the messenger RNA according to the code provided. So the transfer RNA becomes the anticodon. Adjacent amino acids, I'm writing AA there for amino acids, adjacent, meaning those amino acids next to each other, combine by dehydration synthesis to form peptide bonds. Okay, I hope we got that. When this happens, obviously, two amino acids joining together will make a dipeptide, 3 tripeptide, many more than that, polypeptide, and more than 50 amino acids will form a protein. So in this way, this process carries on. I'm hoping you saw how I wrote one point at a time, one following the other, because that is how the marks are allocated. And notice that when you're saying it this way, you are trying to make it in a logical sequence so that you do not get muddled up in your head. Once you start writing big compositions, you forget what you wrote there and you start repeating yourself. So please make sure that you try to keep the essential points clear so that the examiner, when he's marking, it becomes easy for him to pick up your answers. Obviously, our last point here will also be that this process is controlled by enzymes.
Yes. Thank you. I hope you guys had time to jot all that down because this will help you for your exams. Before your really quick break, I want to remind you of what prizes we have in store for you. We have CDs, 2 awesome CDs. This one is the YFM one, and this one is Robin Thicke one. We also have this amazing Mindset t-shirt that I'm wearing. Let me show you. Amazing Mindset t-shirt that I'm wearing. And we have the revision book as well that you guys can win when you answer this competition question. It will be repeated during your break. See you right now. Welcome back, Mindsetters. Hope you guys had a lovely, rejuvenating, short break. There is a lot to go through, so you actually don't need breaks. So, Belinda, stop giving them breaks because we have so much to do in the show. I want to remind you guys that you're allowed to ask any questions on the event wall as well. If something is confusing you, if something in paper 1 or DNA and RNA, it's not really sinking in, let us know where you are stuck so that Aslam can answer any questions that you have. Right, Aslam?
There you go. We're waiting for that question.
Sure, we're waiting.
Can we go? Right, guys, you had your break. I just left the question again there for you for the competition so that you can see it. And we go back straight into the next question. And this question deals with DNA fingerprinting or DNA profiling. And the question is also from March 2010. paper 1, question 2.3, the very next question. Study the diagram below which shows the following DNA profiles or genetic fingerprints. What do they show off? The blood of a raped female victim, the blood of a raped female victim of 3 suspects, and semen that was found on the female victim. So obviously this was a rape victim, hence we are looking at the DNA that's found at in the semen as well. And they have given you the DNA profiles of these various samples that they have taken. The semen, the victim, suspect 1, suspect 2, and suspect 3. These were from their blood samples. And this was taken from the semen that was found inside the woman. The first question says, which suspect was most likely to be the rapist? Most likely to be the rapist. Now here, people, you've got to look at the codes. And let's go back there. There's a semen that was found on the woman. Here's the victim sample, and these are the suspects. We look at suspect 1, and we look at the codes, the barcodes. Remember, people, these DNA profiling or DNA patterns that they're showing you are very similar to the barcodes that we see every day when we go to the store. You go to any of the supermarkets, you buy a lunch bar or any other chocolate or any cold drink, you go to the till, they scan the item, whatever you have in your arm, in your hands, would obviously then show up on the screen with the price. Further than that, they have other roles where they can take off that off stock and so on. We're not interested in that. But if you take a particular item A and they scan it, it's not going to give you the price of item C, because each item has a specific code. In the same way, you and I, each one of us, we have a unique identity code inside our body called a DNA fingerprint, which you cannot change. So that's what we are looking at. So let's look at this barcode. If we had to scan this barcode for this semen sample, obviously it's going to give us a negative result because this one does not resemble that one much. This one here, suspect 1, would obviously not match there because the bars do not match. Let's look at suspect 2. Suspect 2, same situation. You can already look at the top. It's very different here. Go at the bottom. It's different there as well. And that leaves us with suspect 3. And if we look at suspect 3 and we look at this bar, it's almost an identical match. There is, I think here, just because of the duplication of the paper, what happened is this may have moved out a little bit. But generally, if you look at this bar and you look at this bar, they are identical. So the first answer there then would be suspect 3. Suspect 3. And obviously, guys, the minute you're going to make a choice, reasonably sure you can be that the very next question is going to ask you, why did you choose that particular one? So there's it there. Explain your answer to question 2.3.1. And this answer, this second answer, will never change generally in a crime scene especially. It doesn't matter which suspect you had chosen there, the correct one obviously. Your answer will always be because. I was very tempted to write because the way you write it on your text so you understand it a bit better, but I changed my mind. Because suspect 3 has a DNA profile that matches that of the semen. That will always be the answer. Whether it's semen, whether it's blood, whether it's hair, your answer would be that the DNA of the suspect, the one that you choose, matches the DNA of the sample found at the scene of the crime. So that answer doesn't change. So please keep note of that. Give two reasons why this evidence may not be considered reliable. Here's evidence. They're showing you the semen that we found on this woman or in this woman is exactly the same as suspect 3. So you are in court and you're arguing that this is the story, that this person should be charged or found guilty of murder. The question is, why is this not totally reliable? It doesn't say totally reliable, but why is it not reliable? Yes, you got it? Because the evidence could have been planted at the scene. Not a very good choice of words in this particular case, but you get my point. I want to use this word because you must understand what we are saying here. This could be placed at the scene. Yes, even semen can be placed at a particular crime. Remember, people use condoms, etc. They could collect that semen and drop it off anywhere they want to. Or, so at the scene is one. Secondly, this person, the suspect 3, could be in a relationship with this woman, and he could have been having sexual intercourse with her, and obviously the semen could still be there when being investigated. Remember from your reproduction that you learned in paper 2, semen has a lifespan of plus minus 72 hours. Sperm cells can last for up to 72 hours. So obviously that is still there. Okay? So in essence, what we are saying, if we take these 2 points, we are saying that yes, this DNA sample tells us that this particular man was there. He was at the scene of the crime because we found his semen there. So, but somebody could have planted it there, one, or he could be in a relationship. So it does not prove conclusively that he raped her or that he killed her, because he could be in a relationship with her, etc., etc., etc. Good? Let's move on. Name 2 benefits of DNA profiling other than for solving crimes. Okay, so they've given us one benefit already. One advantage of DNA profiling is that it can help us solve crimes. Now, most of you don't read this part here. Other than solving crimes, and your first answer is to solve crime. No, you can't do that. 0, no marks for that. Okay, so the others are, one is health issues. It can help us to cure diseases. If we learn or if we find out the DNA code for a particular virus or bacteria, etc., that is causing the mayhem in the body, then we can develop medication or cures to deal with that particular issue. That's one. The other one is, and the name that comes into my mind every time I talk about this, is a person like Michael Jackson, and that is paternity issues. In other words, when we want to prove who the father of a particular child is. And sometimes we can even go further than this, than to prove relationships also in immigration matters and so on. Paternity issues meaning we want to take the DNA of the child, we want to see whether the father's DNA has any matches with the child. Remember, however, when it's a paternity issue, you cannot have a perfect match because the child's DNA has DNA from the mother and the father. So obviously the child's DNA is his unique DNA. It's not exactly like the mother's, nor is it exactly like the father's. His DNA or her DNA is unique to them. It's a mixture of both. So obviously in those issues, you're not going to get an exact match, but we look for similar patterns along the way. So paternity issues is the other one. Oops. Okay, that covers us with the question on DNA profiling. That question comes like that most of the time. Or it can even come with a graph with similarities, etc. So please make sure you learn that section and you figure out how you pick those answers. We move on now to question 3.2 from the same paper, and this one here talks about a table below which shows the relationship between the age of a mother and the risk of having a Down syndrome baby, a baby that is suffering from Down syndrome or is affected by Down syndrome. The table is very clear. On the left column, we have the age of the mother in years. Notice how they're drawing the table. They're giving you the characteristic or the heading, column heading, with the units. And on the other one, risk of Down syndrome baby with the units per 10,000 births. Okay, and the rows are drawn very nicely. I'm stressing this because you are also asked to draw tables in your exam, and you need to make sure that these tables are drawn in this way. You must have a heading for the rows and the columns, etc., and you must rule off, etc., and have a heading if you need to do that. This one says, state the relationship between the mother's age and the chance of having a baby with Down syndrome. So we want to know what's happening with this part here and how is that related to this part there. What is the relationship between these two things? And as we can see, as the mother's age increases, so too does the risk of having a baby with Down syndrome. In all these relationship type questions, you get into the habit of saying your answer, or writing your answer rather, I should say. You don't say your answer, you write your answer. You write your answer in this fashion. As, you start with as the age. So you're dealing with one column, age of the mother increases, so too does the risk of having a baby with Down syndrome. Or you could say it in your own words, the chances of having a baby with Down syndrome increases with age. Still a relationship. How many times, by how many times does the risk of having a baby with Down syndrome increase between 35 and 45 years of age? Now notice, it said by how many times, It didn't say how much more. It says by how many times, Let's go there. 35, it was 25, and 45, it was 200. So, at 45, 200, and 35 was 25. Now, many learners, what they do in this situation, they say 200 minus 25. And they give an answer of 175, which is incorrect, because they want to know by how many times. By how many times? Multiply. So what do you do to get to that? You divide, you go the other way around. So it'll be 200 divided by 25, and you say 8 times more. In this particular question, they only gave 1 mark, so you just had to say 8, it was good enough. If they said 2 marks, if they gave 2 marks, they would have given you the second mark for that unit on the other side. Always be in a habit to write the units, it's easier. How many chromosomes are present in a cell of a body of a baby with Down syndrome? That's easy, 47. Straightforward question. The last question says, explain why a person with Down syndrome has an abnormal number of chromosomes. This particular question, Pat, we will deal with immediately after the break.
Okay, awesome. In case your brain is fizzling out, Aslam is giving you a very, very quick break to get refreshed. See you after the break. Welcome back, Mindsetters. Hope you guys had a great break. I can see all your answers are streaming in, and I'm loving it. And also, I'm loving that a few clues have been placed in these lessons. So I'm sure your guys' answers, wherever they are and however they are on the mark. Hey, Aslam.
Yep, there. Are we
Take
ready
it
to
away.
move?
Yeah.
Good, guys. We stopped at this particular question here. Explain why a person with Down syndrome has an abnormal number of chromosomes. But before I tackle this question, I want to go one step back. One of you gave me an alternative answer for this one here. Let's go back. So, on the profiling issue, I'm just going back to that one. State 2 reasons why this evidence may not be considered reliable. One of you posted on Facebook that could the answer be that this person has a twin? If you wrote that in an exam, a person has a twin on its own, that would be marked incorrect. It has to be an identical twin. So the person may have had an identical twin. Very good answer, but remember, you have to use the word identical. It's only identical twins that will have the same barcode, if I may call it that, the DNA profile. Why? Because they come from one sperm and one egg. They're identical. The word identical says it all. And by the way, many of you, I get this question often at school and other places. They say, can a brother and a sister that look the same and they were born at the same time be identical twins? Come on, guys, look properly. You're not looking closely enough. Look all over. You'll note that they definitely can't be identical. Okay, so look properly. I don't say take anything off. I said look properly. Okay. Let's move on. Go back to our question. And we were dealing with this one here, the last one. They say, explain why a person with Down syndrome has this abnormal number. And we've already said what this number is. We said that it is 47. In other words, this particular person has one extra chromosome. How? How did this happen? And our answer starts by saying, during anaphase of meiosis. You can say anaphase 1 or anaphase 2. They are both correct. It can happen during anaphase 1 or anaphase 2. So during anaphase of meiosis, or rather, let me just change that to one set of chromosomes do not separate. And obviously, guys, the result of this would be that one cell is going to have a chromosome too many. So in other words, one gamete will therefore have an extra chromosome. If this gamete, which one? This one that has the extra one, fuses with a normal gamete. Normal gamete in this case meaning one that has 23 chromosomes. Then the resulting zygote would have 47 because 23 plus the one extra from the other one, 24, gives you 47 chromosomes. So that is how we answer that question. Again, notice I'm trying to put one fact per line. In that way, you can see how the answers marks are allocated. Very important to see that. Are we good with that? Pat, you had a question.
Yes, there is a question. This question is by Mokonazwote, another name Zoe. Can we call you Zoe? Thank you. She asks, she had struggled with naming the differences between DNA and RNA. 5 differences.
Okay, we're going back to that, to DNA and RNA. We said, first of all, DNA is found mainly in the nucleus, and it may be found in the mitochondria. If you learn your evolution, you'll know that we talk about mitochondrial DNA as well. Whereas RNA is found in the nucleus and the cytoplasm. That's in terms of where we find them. But now you must be careful about the question also. The question may ask you structural differences. If they talk of structural differences, then obviously this one is out. You can't talk about this. This is talking about where we find them. Now let's go to the structural differences. Let's look at firstly that DNA is a double strand. RNA is a single strand. Secondly, DNA has the nitrogenous bases adenine, guanine, cytosine, and thymine, whereas RNA has adenine, guanine, cytosine, and uracil. Okay? That's the third difference. We also know that the sugar that is found in DNA is deoxyribose sugar. Deoxyribose sugar. And in RNA, it's ribose sugar. In terms of the chemistry of this, it simply means that the deoxyribose sugar has 1 oxygen less, deoxy, 1 oxygen less than that one there. It's actually, if you just put it there, this you do not need to know for the exams, C5H10O4. It should be 10 and this should be 5 as well. And this one will be C5H10O5. So here we are short of 1 oxygen. I'm not going to go into the chemistry of that because you do not need to know that for this paper. I'm simply just explaining the difference between deoxyribose and ribose sugar. We go further, my line is not so straight, and we talk about the types of DNA. There's only 2 types of DNA, and that is in the nucleus, the nuclear DNA, and the mitochondrial DNA. That's the 2 types of DNA that we speak about. Whereas we have 3 types of RNA, and that is messenger RNA, transfer RNA, the 2 we've spoken about, and also we have ribosomal RNA. There you have your table with 5 differences. I'm hoping that answers Zoe's question.
Yes, right? Zoe's question.
Okay. Good. We now move to question 4.4, which is an essay question. And this question appeared last year, November, NSC, paper 1, question 4.4. And the reason we've chosen this particular question is so that we can get used to answering essay-type questions. We find in exams many learners mess this section up because they do not know how to organize the essay. This essay in life science is a factual essay, not like composition in creative writing in English or any of the languages, or your essay in history for that matter. This is totally different. Here we want you to tell us what the question is asking you to do. And let's see what we are saying there. The first thing that we look at in any question for that matter is the verb. And there's the verb. Describe. What does it ask you to describe? It describes the mechanisms by which meiosis contributes to genetic variation. And describe how abnormal meiosis, another describe, leads to Down syndrome, and polyploidy. And also describe the advantages of polyploidy in agriculture. So can you see that there is 1, 2, 3, 4 parts to this question? That's the first thing you needed to ascertain. What do I need to answer? I got 4 things to do here, and I need to do it that way. And the easiest way to answer a question like this is obviously, guys, obviously, follow the order in which the question is asked. The first describe says, how does meiosis lead to genetic variation? The second one says, describe how abnormal meiosis leads to Down syndrome. The third one says, describe how abnormal meiosis leads to polyploidy. And the last one says, also describe the advantages of polyploidy in agriculture. So there are 4 parts to this question. So let's get into the answering of that question. So the first one, and I'm not going to rewrite the entire essay here, guys. That would take forever, Pat. We won't make that. So we're going to break it up into headings, and I'm just going to put bullets so that we know what we need to discuss. So the first one was meiosis and variety. You can even use this as a heading in your essay. There's absolutely nothing wrong with it. You will not be penalized at all. So you can write that down as a heading. So that's the first one. And what are we going to discuss here? We're going to discuss 2 important issues in meiosis. One is crossing over, and the other one is random assortment. Crossing over, we're going to discuss in detail in one of the questions that we have. So I'm not going to go into too much of detail there, and I'll go back to random assortment just now. The second one says, how does abnormal meiosis, Down syndrome? So the next heading is, you can even write there, meiosis and Down's syndrome. This one, I don't have to discuss with you again, guys. We just discussed this already. The same answer we had above just now, that answer fits in here. The same thing. During anaphase, the chromosomes do not separate. One set of chromosomes do not separate. And by the way, sorry, in that answer, I'll have to go back to that. It's not just one set. It's chromosome pair number 21. Chromosome pair number 21. You must specify that. I'm sorry I didn't mention that earlier. But that we talk about chromosome pair number 21. Good. The next one says polyploidy. Now, the way you answer polyploidy is very similar to this one. However, in polyploidy, we talk about entire sets of chromosomes that do not separate. Where this is aneuploidy. This one here on Down syndrome, because it's dealing with one particular set of chromosomes. Here we're talking about polyploidy. The entire set of chromosomes do not separate during anaphase. Therefore, one gamete would have diploid instead of being haploid. And if that particular sperm fuses with an egg, or pollen grain fertilizes an ovule, then obviously 2 plus 1 gives you 3. So that would be triploid. So that's where polyploidy comes in. And the last one says advantages of polyploidy in agriculture. So there's our plan. This is what we have to do. We need to add meat to this. This you could have done on your question paper very roughly, or even on your answer book, and then you scratch it out neatly as a plan. You do not have to show a plan in the answer book itself. But obviously the way you set your answer out tells us whether you planned this thing at all. Because if you planned it, it's going to follow the sequence. If you did not plan it, it won't follow the sequence. I'm hoping that we got that, and this is what we're going to discuss there. In terms of random assortment, we talk about in metaphase of meiosis, where the chromosomes are in no particular order, like the lotto balls. There's no order. Nobody can tell you next week or tonight that it's going to be this number. It's a random process. So random means there's no order in which the chromosomes are lying at the equator. So when they separate, they can separate in any order. And because of this, every time this meiosis takes place, the cells that are resultant would be different. With that, I give it to Pat.
Thank you. Guys, we're going to go for another quick break. I see there are plenty of questions. I'm going to give them to Aslam during the break so that he can see which one to answer for you guys. See you after the break. Welcome back, Mindsetters. I've been informed to tell you that the competition is now closed. I hope you guys have sent in all your answers with the unique code in our events or pep text or email, or you've SMS'd it or you've called. That's it. The competition is now closed. So we will be announcing the winner at the end of the show so that we can celebrate how they have won. Over to you, Aslam.
Thanks, Kat. Guys, I'm going to use something that I've made a mistake with to explain something very important. And that's about mutations and protein synthesis. And you remember you have let's use what I've made a mistake with. Let's say, for example, one of the nitrogenous bases was C. Where is that? C-A-T. That was a nitrogenous basis. And I mutated it, and I changed that to P. So I've been calling Kat Pat all the time. She wasn't that upset because it didn't make a big difference to her life, did it?
No, not
Not
really.
at all. Okay, so this is what we call a point mutation, where one of the nitrogenous bases actually changes for another one. That's a point mutation. A frame shift mutation is when, if we take out one of these, let's say we had A, C, G, T, C, and for some reason we take this one out. So whatever reads after this is going to change the sentence. It's going to change the meaning of this word. And if the meaning of the word changes, then obviously the type of code that it's sending out for the amino acid will also change. And if that changes, then the wrong amino acids will get together, and this will change the structure of the protein. And because the structure changes, the function of the protein will also change. So please understand that. Sometimes in this code of DNA, this beautiful code of DNA that God has given us, sometimes mistakes do take place. And the two main types of mistakes that you must concentrate on are the point mutation and the frame shift mutation. The point, one base is replaced by another one. The frame shift is either this one here with deletion. Let's say I had T there. There we go. I had T there. But also sometimes instead of a deletion, that was deletion, another thing that can happen is somewhere in between one squeezes in. That is called an insertion. But already can you see that there's three bases now? And each of the three, each set of three, actually I should say, each set of three is a gene. And that is what is coding for the transfer RNA eventually, 3 at a time. We call them base triplets. So see what has happened there. We had TC, now it has become TAC. So obviously, if this was DNA, then the messenger RNA would have been different. And if the messenger RNA is different, then the transfer RNA is also different. So this is in terms of mutations. Let's move on to a question on meiosis. The diagram represents a process taking place during meiosis. So they're telling you that it's meiosis. They didn't ask you to figure that out. They've given you a beautiful diagram there, and they've given you some labels, diagram representing a process taking place during meiosis, etc. Say provide labels for parts A, B, C, D. As usual, I would label on the diagram so it makes it easy, so you don't have to keep flipping through. A is referring to this fellow only, alone. And you can see not only one of the two strands, but the whole of the story there. So that would be a chromosome. Chromosome. Okay? B is pointing to this little circle in the middle there that's holding these two strands together, and that is known as the centromere. C is pointing to one strand only, and that is the chromatid. And D is pointing to an area there. This area where the chromosomes are overlapping each other. And because it's only talking about one, we will label it as the chiasma. Plural for that is chiasmata. When there are more than one, it is known as chiasmata. We are talking of the chiasma only. So there's our labels, guys. A were the chromosomes, B centromere, C chromatid, and D the chiasma. Let's move further. Name the process in meiosis that is illustrated in the diagram above. Now notice very carefully, they're asking you for a process. They didn't say which stage of meiosis. They did not say which phase of meiosis. They said what process is taking place here. And this is where many of you get it wrong. Immediately, you'll either say it's meiosis 1, which answer is incorrect. Some of you would say it's prophase. Your answer is still incorrect. Although, your answer is not incorrect completely, but it's not answering the question. The question didn't ask you for the phase nor the stage. It asked you for the process. And the process that's taking place here is this process here that we are talking about. And that process, guys, is obviously crossing over, which we spoke about earlier when we spoke about the essay. State one importance of the process mentioned in 212. And I'm going to go further with this. I'm going to say, let's talk about the significance in general. What is the significance? What is the importance of this process? Crossing over, what does it do? First of all, crossing over allows for exchange of genetic material. That's the first thing. That's what's happening. Let's go back to the diagram. Here's these things. They're crossing over. And when they overlap, at that point, some of this chromosome is changed with some of that chromosome. So some genes are going from chromosome A to its partner, to its homologue, we would say. Remember, together, this represents, if we had a bracket this way, then this is known as the homologous chromosome. So we're saying that this one is exchanging with that one. So let's see what we're talking about there. So first of all, allows for genetic material exchange. So exchange, one mark, genetic material, another mark, depending on the mark allocation. In this case, it was only 2, so that would have been good enough. Or we carry on further because I said we want to talk about the general significance of this. So we're saying allows for exchange of genetic material, which, and this is important, brings about variation. So if it was 3 marks, then your third mark would have come here. Because of this exchange, we are swapping. From one, something is going from here to the other one, and something from there is coming. Obviously, after this process, these two things are not the way they were before. They're different. And what is different? Variation, variety. So this is what meiosis, one of the most important significances of meiosis is exactly this. That because of meiosis, there's differences among organisms. You and me, look at your own family, brothers, sisters, etc., in your own house. You all look similar, but mom, dad, and maybe even the school teachers and the neighbors can distinguish between Aslam and Lebohang and Kat and Tracy, etc., etc., etc. Because we have distinct differences brought about because of this crossing over. One of the reasons for variety is this one. This whole section here, by the way, would have gone there by the essay when we discussed crossing over. This is how you would have discussed it. That is why I'm taking more time there. Okay, good? Let's move on. During which phase of meiosis does the process named in question 212 occur? I must tell you at this stage that currently the examiners cannot ask you which phase. But I always find it easier if you know the phase, because you cannot explain about crossing over. You don't know this happens during prophase. But in an exam, they wouldn't ask you which phase. So I'm going to change. All right, let's answer that first. Obviously, if it's crossing over, then it is prophase 1. Now I'm going to change that. Let's say, take that out. During which stage of meiosis does this occur? Then your answer would be, and this they can ask you still, meiosis 1. So in the current syllabus, this, remember this was for 2010 March, it was taken out from last year 2011 November, and now we can say meiosis 1 in that particular case there. Draw a diagram of the structure labeled A to show its appearance immediately after the process. We're going to go to the diagram. Here's the diagram. They want to know this guy here. How is it going to look after this crossing over? So we keep that part of the diagram. Remember, you're going to draw it in pencil. So that's the first part, and we know that it is shaded. You're going to shade it with a pencil. Nothing happened here. There was no crossing over here. The centromere still stays there. Not worried about the color of the centromere. This bottom part you draw again. So you show that is still shaded. Nothing happened there. Now that's where we start. We're going that way. There's that part there. Now that part is still shaded. Then we have a small portion. Sorry about that. That will be unshaded. And then you'll continue with your diagram and you'll have a shaded portion. So you will leave a portion, this portion, unshaded, to show that crossing over allowed one part of this chromosome to come there. That's all you needed to do. And obviously, people, if you draw a diagram in life science, you have to label it. So chromosome 1 or chromosome A after crossing over, that will be your caption. This will be a chromatid, this will be a chromatid. Centromere you can label, and you will say portion that was crossed over. Let's see how many marks they gave you. They gave you 2 marks. They were mainly worried about the diagram itself and the caption. So first of all, it must be a reasonable diagram of a chromosome, and secondly, you must show that crossing over has taken place there. Okay.
Aslam, Mbali or Mbavi, asked, does meiosis contribute to cancer?
Good question, Mbali. Does meiosis contribute to cancer? Not so much meiosis, but generally any cell division can contribute to cancer. What is cancer? Cancer is cell division gone wrong. Remember, we have 2 types of cell division, mitosis and meiosis. So both of them can go wrong, and when they go wrong, the cells don't know when to stop dividing. And when they carry on, generally, however, cancer is restricted mainly to mitosis. When the cells are dividing, for example, as we're speaking, we're losing skin. Skin cells keep dividing to make more skin, and so your skin gets replaced all the time. So if those skin cells, for some reason, some mutation took place, some code went wrong, and they don't know when to stop. Where's the full stop at the end of the sentence? I must stop here. Then the cells will continue dividing all the time, and you'll have skin cancer.
Yes.
And so on, lung cancer, whatever. What causes the cancer? It's not the mitosis itself, but it's triggers, which we call carcinogens. Anything that causes the cancer is known as a carcinogen, and that will trigger it off. What are those triggers? Smoking. I put that one first because at your age, you're already experimenting, or if not, you've already hooked. Smoking, exposure to sunlight without protection, skin cancer, and at the same time, you can be exposed to chemicals in your environment, and the cell phones can also cause that. Guys, I'm hoping that what I've done now has helped you for this section up to meiosis. And with that, I hand you over back to Kat.
Thank you, Aslam. Guys, we're going to go for a very quick break. After this break, we'll be going through this competition and who the winner is. See you after the break. Welcome back, Mindsetters. I'm glad to see the interaction on the page. You guys are writing a lot today. I know life science is very exciting. I love it as well. That's how I know that you guys are loving it too. Now we're going to answer some of your questions. So if you still have questions, send them through as fast as you can, like in the next 2 minutes, and we might just get to them. And we'll also be answering the competition question for you right at the end. So the first question that you want to answer, Aslam, is what is polyploidy and what are the effects or the advantages on agriculture?
Thanks, Kat. So the first question is what is polyploidy? I'm just going to write polyploidy there. You'll know what we're talking about. And what are the advantages? And if you know, guys, remember this was the question we asked in the essay. What are the advantages of polyploidy in agriculture? These are the two questions that we're going to deal with immediately. First of all, polyploidy, how do we explain this? We explain how it happens. We need to know now what it is. Polyploidy is a condition where an organism has more than 2 sets of chromosomes. Always better to delete. That's what polyploidy is, first of all. In other words, instead of being diploid, the organism has more than one set. Could be triploid, tetraploid, and pentaploid, and so on. But anything more than 3, polyploid. Okay? So in other words, this organism is supposed to, let's take humans for an example. We have, how many chromosomes can you remember?
46?
Hey!
We're
giving her the prize. That's why she's wearing the t-shirt
that's why
already.
I have a t-shirt.
There we go. 46 we have. So in other words, every human being should have 46 chromosomes. We saw what happens if just one chromosome was added in Down syndrome. Just one chromosome, and that's only one example of chromosome number going wrong. There are many other examples that are prescribed and are in your textbooks you can look at. But Down syndrome is what we discussed here, so we can talk about that one as well. So you can see what happened. Now imagine if we had 46 plus 46, 92. Each species has a particular number of chromosomes. And the number of chromosomes does not mean that you are more advanced or you are less advanced. For example, we have 46. Ferns, some ferns have 300 chromosomes. But look at the ferns, they're much smaller than us. And we think, humans think, that ferns are less advanced than us. We don't know. We assume that we are more advanced than the ferns. But they have 300 chromosomes. Now, can you imagine if we double that, what can happen? Okay, so that is what polyploidy is all about. And how does it come about? Is when the whole set of chromosomes do not separate during meiosis. Remember, during meiosis 1, the homologous chromosomes separate, they move apart. And in meiosis 2, the chromatids separate. So whether it's anaphase 1 or anaphase 2, doesn't matter. If those chromosomes do not separate, then the result cell or gamete that is formed there would have a whole set instead of half. And therefore, when it fertilizes a normal one, 2 plus 1 will give you 3, triploid. Or if there are 2 of them, 2 plus 2 gives you 4, tetraploid, and so on. And if these carry on that way, then you can get octaploid. Some strawberries, by the way, delicious strawberries. It's Wimbledon time now. And when we talk of Wimbledon, we only talk about strawberries and cream. So some strawberries that we eat are octaploid. Okay? What are the advantages? That brings us to the next part. What are the advantages of polyploidy in agriculture? First of all, we can get more crops. In other words, a greater yield. And this, people, it can't stand on its own. Why do we have a greater yield? We're having a greater yield because we get sometimes because of my head is working faster than the pen. So we have some that we form are pest resistant or drought resistant. Sometimes we can get this situation, and that can help us to get a greater yield. In other words, what we're saying here, you'll see some of the textbook talk of a hardier crop. Hardier means it is stronger, it can last longer. So why? Because it resists pests and drought and harsh conditions. So that's one advantage. So we get more crops, a bigger yield, etc. The other advantage, in terms of the strawberries that we were talking about earlier, we can get bigger fruit. So we can get bigger fruit. And obviously, economically, this is a great thing. If you're getting bigger fruit, people are going to take those. And therefore, they're going to pay maybe more money for that, etc., etc., etc. And obviously, apart from the fact that eating a bigger fruit is going to last you longer, and therefore, it's going to be tastier for a longer period of time. And in your house, if you buy only one punnet of strawberries, I know in my house, they'll fight for it, then maybe you'll be able to manage to share that, especially if you have a big family. Okay? Bigger fruit, we can even have bigger seeds. And we also have seedless variety. Seedless variety of fruit. These are some of the advantages. We can also talk about increase fertility. I have also read in a study in United Kingdom where it can also decrease fertility. Polyploidy can also decrease fertility. But we are looking at advantages in this case here. But in that case, it was an advantage. I do not want to go into that because it's not prescribed in a syllabus. These are the advantages that are prescribed in a syllabus, and you rather concentrate on these. If you want to, you can do a Google, infertility plus polyploidy, and you will come to a section that talks about grass carp. It's a type of fish. In the United Kingdom and in America, you can read about that if you want to. That's for if you want extra. Right now, we want to know about what we need to know for our exams. That's first. Okay, that answers that question. The next
Do you
one?
want another one? Thank you, Tebuko and George. Those were awesome questions. The other one is, what causes mutation and what are its effects on protein synthesis?
So we're talking about mutation. The two things you want to know are the cause of mutation and its effects
on protein synthesis.
on protein synthesis. Good question. This second part especially has appeared in many essays where they ask you about the effects of mutation on protein synthesis. But we start by explaining what a mutation is. What is a mutation? A mutation is any change. If it is a gene mutation in the DNA or in the gene. That's the story there. Any change in the DNA or gene. If it's a gene mutation. If it's a chromosome mutation, then it'll be any change in the chromosome. Number of chromosomes, size of chromosome, etc. That's the first thing. What causes this? Now that, we could have had another 1.5 hours just to discuss the causes. I alluded to some of them earlier, that mutations are caused by smoking. Whatever caused the mutation in cancer, those same things can cause mutations in the DNA as well. But also some mutations are so-called natural. They take place on their own. And some of these mutations are the result, or rather, the result of the mutation is why some organisms are so different. And sometimes mutations could be beneficial to an organism in such a way that that organism has a better advantages than those without the mutation. Let's say in murky water, fish were all white. And all of a sudden comes one black fish there. Okay? And when that happens, that black fish has more chance of survival than the whiter fish because they can be eaten quicker by the predators. I'm just giving you a wild example here. So that mutation was beneficial, and those organisms will survive, the others will get wiped out. So that's there. The last one is the effects of mutation on protein synthesis. This is the easy part. I explained this when I spoke about cat and pet earlier, that when a mutation takes place, obviously if the mutation is on the gene, this is on the DNA, obviously. And if a mutation takes place on a gene, then this gene is going to code for the wrong messenger RNA. Look at the steps in this answer because you can get a mark for everything you're talking about. The wrong messenger RNA, and that in turn will code for the wrong transfer RNA, and that in turn will code for the wrong sequence of amino acids. And if the sequence of the amino acids are different, then the protein formed would be different. That answers that question, Kat.
Yes, it
and
does.
Now,
the
the
competition
moment
question.
has arrived.
Yes,
So over
let's
to you.
go through it. Let's
There
go
we go.
through the competition question. Do you have
Let's
it
go
there?
back to the question, yes.
That's it.
In DNA, thymine always pairs with adenine, uracil, or guanine. And guys, I have answered that question more than once today. Over to you.
I can guess. Let me guess. Actually, I won't guess because I actually know the answer. The answer is A,
Answer
adenine.
is A, adenine. Why? Because, first of all, in DNA, there's pairing. That's the first thing. So, obviously, there's thymine. And remember, we always use the letters at. Adenine combines with thymine. And cute girl, as I told you the last time, cute girl, cytosine with guanine. If it was RNA, then obviously we cannot talk about pairing as such. We can talk about matching, but not pairing, because there's no pairing in RNA as such, because it's a single strand. So uracil is definitely out because it is not in DNA. So you are left with these two, and using these two simple mnemonics, at and cute girl, you would have sorted it out.
Thank you, Aslam. Unfortunately, we cannot announce the winner for you because it was an SMS winner. So if you happen to SMS us the answer today, maybe just try sending your name and your address and everything. Let me check again, actually, in case I can actually find it. Nadia, that's you. You are the winner. Nadia, you win the CDs.
Well done,
2
Nadia.
CDs. You win the revision textbook, and you win this awesome Learn Extra t-shirt. And guys, from us, Aslam and I, we wish you very well during exams and everything that you may have, and also enjoy the rest of your holiday. Bye guys, see you next time.